Hello, welcome to 13 Universal. This is A-level Caie biology, and we're looking at nucleic acids and protein synthesis, MCQ, paper 1, P1, of course, series 4, in preparation for your next exam, which is on the 11th of June 2024. So this is Shedro, your teacher. Come on, let's go and solve some questions. Number 1 says one characteristics of DNA, okay, is that it is a universal genetic code. Now that is genetic code, all right. So what is meant by universal genetic code? What does it mean? So these are the options. I would like you to pause the video and try the question. Universal genetic code. A says all living organisms use the same triplet code for amino acids. All DNA triplet code for different amino acids. Not all DNA triplets code for an amino acid. All living organisms contain the same 4 nucleic acid. What do you think is the answer? Okay, so I think the answer should be A. Okay, now why? Because universal genetic code means that if an amino acid is coded by a particular base triplet in one organism, the same amino acid is coded by the same base triplet in all living things, whether it is in human cell or plant cell or fungal cell or bacterial cell, the same. Is that okay? So without wasting time, so all living organisms use the same triplet code. That's universal genetic code. Number 2, which statement about mRNA is correct? Okay, pause the video and try it out. Which statement? So A says it is a polymer made of nucleotides all joined with hydrogen bonds. Now hydrogen bonds is not the only bonds that join nucleotides. Okay, well, hydrogen bond only joins the bases on the nucleotide. Okay, and of course the bases on one nucleotide and the base on another nucleotide. Is that okay? So but mRNA is a single-stranded structure. Okay, so it's less likely to form hydrogen bonds. Is that okay? So this is not correct. It's single-stranded. Yeah, it's single like this. And of course, if we have this strand, we have A here. Okay, we have maybe C. These are bases, right? This is D. No, there's nothing called D. You have G. Okay, there's no base called. We have G. That's G, right? G. And then we have maybe C again. T. You see, so it's single-stranded. So it's likely going to form hydrogen bond, right, with itself. Except it's going to form hydrogen bond with transfer RNA or DNA. But if DNA of course is double-stranded, so likely form hydrogen bonds. So that is why for messenger RNA, A is not the answer. B says each nucleotide subunit contains sugar ribose. Yes, the sugar, pentose sugar found in mRNA nucleotide is ribose. Okay. C says it always has an equal proportion of adenine and uracil. Well, that's not correct because it's single-stranded. But if it's double-stranded, that would have been correct. If it's DNA, that would have been correct. So this is wrong for a single-stranded structure like mRNA. This says the mRNA sequence is identical to the template of strand of DNA. It's not identical, it's complementary to the template strand. Okay, so B explains it well. Okay, so the diagram shows part of the process of translation. So that's it, guys. I'd like you to try this question. Okay, what are the names of the structures labeled X, Y, and Z? What are the names? Okay, so let's go to pause the video and try it. So this is X. So X, this is transfer RNA. So Z is transfer RNA. Okay, therefore X should be anticodon. So X is anticodon. That eliminates, all right, C and D. Okay, because X is found on transfer RNA and that's anticodon. So let's go to, of course, Y is codon, all right, because this molecule is mRNA. This particular molecule here is mRNA. It's acting as a template. So that's messenger RNA. Okay, so the base triplet or the sequence of 3 bases on mRNA is Y, and it's called codon. So codon, Y is codon. So, and of course the entire structure Z is transfer RNA. Okay, that makes B correct, and of course A is wrong because this Z is not mRNA. mRNA is this. Okay, all right, so the answer is Mr. Bravo. Okay, let's go to the next question. The DNA triplet of genes are translated as amino acids or stop signals during protein synthesis. The table shows some of these triplets. All right, let's look at this. Is a triplet. These are the triplets. You can see DNA triplets and the amino acid they represent or they code. What could be the effect of one substitution in a triplet coding for tyrosine? All right, let's see. Remember that this is paper 1. You have to read the questions critically, read between the lines. So these are the sentences. So which ones are correct? What could be the effect of one substitution in a triplet coding for tyrosine? Now is your turn. Pause the video and try this question. Okay? Pause the video, try the question, guys. Right? So remember, we're looking for the effect of one base substitution. You're substituting with another base. You're replacing a base with another base in a triplet coding for tyrosine. So this is tyrosine. Well, of course, this is tyrosine, right? So you see that tyrosine has different base triplets. Okay. So if, for example, this is a tyrosine. All right. Okay. Let's go to statement first. So the triplet is translated as cysteine. Where is cysteine? Yeah, this is cysteine, right? Yeah. So this is tyrosine. Yeah. If I substitute this T, okay, with C, I'll form cysteine. All right. The same way, if I substitute, yeah, I'll form. So this is possible. It can change from tyrosine to cysteine by substituting thymine with cytosine. Okay. So that's correct. All right. So that means that statement 1 sounds correct. Statement 2 says the triplet is translated as tryptophan. Okay, let's go to tryptophan. This is tryptophan, right? Yeah. So if I replace, yeah, because we're looking at base, all right, base substitution, okay, coding for tyrosine. This is tyrosine. So tryptophan is ACC. Okay. So if I come here, I have ATG, okay. So base triplet on it, which of them give me ACC? Just one base. But you see that I don't think this is going to work. Is that okay? Yeah. So that tryptophan is not going to work. It's going to require 2 base substitution. So I have to change this to, first of all, AC and then ACC. So this statement doesn't sound correct. So let's look at number 3. Says the triplet is translated as tyrosine. Yeah. So if I see tyrosine, it can change from tyrosine to tyrosine. Yes. If I decide to remove A and substitute it with G, I have formed tyrosine. The same way, if I decide to remove G and substitute with A, so I can go from here to here, or from here to here. So that means that statement 3 is actually correct. Now, 4 says translation stops at this triplet. Okay, translation stops at this triplet. Yeah, now that's stop codon. This stop codon, stop signal, right? Okay, can I form stop signal from tyrosine? Now this is ATC. Yeah. So if I substitute A with C, I'll have ATC. If I substitute this ATG, remove G with C, I'm going to have this. So I can actually move this to this, all right, or that to that. Just remove one of the bases, okay, and put C. Okay, you can have stop codon. Any of these, you can remove any of these and put C. So that works. So this, all right, this works. So we're going to have 1, 3, 4. So which one is 1, 3, 4? Looks like it is C. Let's check. Yes, C is the answer. Okay, let's go on. Which statement about messenger RNA is correct? Okay, which statement about messenger RNA is correct? So let's go. So these are the statements. Yeah, which ones are correct? Okay, so check it out. So which statements are correct? Now A says in eukaryotic cells, mRNA is made by removing exons from primary transcript. So meanwhile, you have to pause the video and try it out by yourself. Okay, check it out by yourself, solve this question. All right, but you see, this process A is called splicing. When you are using primary RNA transcript after transcription to form mRNA, but you do not remove exons because exons are the coding. They are the coding. So what you remove is the introns. So this option A is wrong. Is wrong. Is that okay? So you see, option B says mRNA is a single-stranded polynucleotide. That's correct. Containing a different purine base than DNA. This is also wrong. Why? Why do I say it's wrong? Because the two purine bases, which are guanine and adenine, G and A, G for guanine, A for adenine, they are both found in RNA, okay, and DNA. Okay, so this is equally wrong option. Okay. C says messenger RNA molecules contains ribose. Yes, contains ribose joined to phosphate group by phosphate diester bond. Okay. Joined to phosphate group by, okay, that sounds correct. Yeah. So this is correct. All right. Now this says the monomer of mRNA contains phosphate group. Yes. Deoxyribose, sugar, and nitrogen base. Now, mRNA does not contain deoxyribose. It contains ribose. Okay, so D is wrong. All right. So the answer is, of course, C. Next one. Which structures are involved in transcription only? Transcription is a process of using DNA, a segment of DNA, which is gene. Right? We use DNA to make mRNA. Okay. That's transcription. All right. So easy to make mRNA. Just a section. So this is transcription. This process here, right? A transcription. So DNA. Meanwhile, pause the video and try it. I've given you a clue. DNA template strand. Okay. Yes, it's going to be used. So we eliminate C and D because DNA must be used as a template strand. So we are zeroing in on A and B. So let's go anticodons. Now anticodons are found in tRNA. tRNA is not involved in transcription. Okay, so that eliminates A because A supports. Okay. Now B says, of course, B is already the answer, but it's the RNA polymerase. Yes, RNA polymerase is the enzyme that catalyzes this transcription. The enzyme that catalyzes transcription is RNA polymerase, and that makes B the answer. Okay. All right. Yeah. Okay. Yeah. One gene provides the code for the production of which type of molecule? Now this is very simple. Okay. I just said it last time. So see DNA. Pause the video and check the answer. So a gene code for what? That's what they're asking us. A gene is a section of DNA. So this is DNA. DNA has section. From here to here, maybe a section. This section is used to code for what? All right, polypeptide. Is that okay? So a gene is a section of DNA that codes for polypeptide. It doesn't code. A gene doesn't code for amino acid. Okay, it doesn't code for DNA. It's a part of DNA. Of course, it's made up of nucleotides. So it carries the genetic information that codes for polypeptide. All right, let's go to the next one. The table shows some mRNA codons that code for certain amino acids. So you see that? Yeah, interesting. Okay, a DNA template. Can see that sequence shown. That's a DNA template. What would change in the amino acid if, now you have to read the question carefully, okay? If the first base in the fifth DNA triplet was substituted for a adenine base. So remove the first base and put adenine. Okay, these are the options, and I'd like you to try it. Pause the video and try it. That's how we roll. You must solve the questions, okay? So remember, if the first base in the fifth DNA triplet. Okay, let's go. This is a triplet. This is the first triplet. Okay, triplet sequence of 3 bases. This is another triplet. All right, this is another triplet. Okay, this is another. So we're dividing them in triplets. Okay, so divide them in triplets. So this is the first one. This is the second triplet. This is the third. That is number 4. Fourth. This is the fifth triplet. Okay, and this is the 6th triplet. And the question says, what would be the change in the amino acid if the first base in the fifth DNA triplet? So this is the first base here. Okay, G. This is the first base. It was substituted for an A adenine base. So remove it and put adenine. Okay. So they say what would change? Okay. So don't forget that initially that code is GCA, and GCA, that's the DNA template DNA. GCA we code for messenger RNA. This is the DNA, all right, template. This is the triplet on the DNA template. Okay, so now which mRNA codon will it code for, right? So remember, that is a template. So now, of course, GCA codes for, all right, if you, during transcription, G will bond with C, that because they're complementary. C will bond with G, so it's going to be CGA bond with U. So CGU. So let's come here. Which one is CGU? CGU, okay, yeah, CGU, CGU here, CGU, yeah, can see that's the original, right? So CGU is arginine, okay? So, but after the mutation, base substitution, this guy changes from GCA, because A is going to be here, to ACA. So that's going to be ACA on the template strand of DNA. All right. So this ACA, all right, on DNA, what is going to be? It's going to be U, A pairs with U, okay, UGU. UGU is here. Yeah, that's it. So that means that, and that UGU code for cysteine. That means that the effect is that arginine will change. The original arginine will be changed to cysteine. Okay? See? Instead of coding for arginine, all right, the impact of this mutation is arginine will be replaced by cysteine. Okay? Which option? All right? Do we go for C? See that? All right, let's go to the next question. Which statement correctly describes a feature of DNA molecule? All right. So pause the video and try it. Which describes the feature of DNA molecule? If you look at A, it says it has 2 parallel strands twisted into double helix. Well, this would have been the answer, but DNA has 2 anti-parallel. Okay, so anti-parallel. You know, DNA has anti-parallel. Okay, so 5 prime. All right, one runs from 5 prime to 3 prime. The other one runs from 3 prime. Okay, so they are anti-parallel to 5 prime. Is that okay? So that is why this is wrong. Okay. Now, B says it has complementary bases, all held together by 3 hydrogen bonds. Well, some of them are held together by two hydrogen bond. For example, if we have, of course, A forms double bond with thymine, right? While C forms triple bond, right, with guanine. All right, this makes sense, but it captures only 3 hydrogen bonds. Or let's see, C, it has phosphodiester bond. Okay, yes, that are formed using DNA ligase. Now, phosphodiester bonds are not formed using DNA ligase. They are formed using, of course, phosphodiester bonds, because they are formed using DNA polymerase can form phosphodiester bond. Okay, yeah, phosphodiester bonds are formed using DNA polymerase if we are joining it between 2 nucleotides, right? Okay, and but if we are doing between 2 Okazaki fragments, all right, so it's going to be DNA ligase. Okay, ligase form phosphodiester bonds. So, but they say it has phosphodiester bonds that are formed using DNA ligase. Yeah, DNA ligase can actually form phosphodiester bond between Okazaki fragments. Okay, and of course, this also sounds correct. All right. So this says it has a backbone of phosphate molecule and nucleotide. Now, it's a backbone of phosphate and sugar. All right. So we have eliminated A and D. So we have, okay, we have B and C. Which one is more correct, more accurate? So you say it has complementary bases, all held together by 3 hydrogen. Now, all of them are not held together because we're going to have adenine and thymine, and they're held together, right, by double bond, right? So I think C should be a correct answer there. Okay, DNA ligase can form phosphodiester bond. So here the answer should be, it should be C. Okay, you can get back to us if you have questions, but I think it should be C. Yeah, it should be C. There's a typo error. It should be C. Okay, C is more accurate than B, right? So the answer is actually C. C. Okay, yeah, C. Okay. All right, so that is it. So let's go to the next question, guys. Which diagram correctly shows replication of DNA? Yeah, so which one shows replication of DNA? I'd like you to solve this question. Solve it, okay? Remember that replication of DNA, all right, they changed the direction. Before it used to form like this. That's replication flag is like this. Okay, yeah, and normally, okay, we have this 3 prime, okay, to 5 prime normally, and then we have 5 prime to 3 prime. Okay, so this is the direction of winding process, and this goes in the same direction. So this replication here copies leading strand. Remember that S1 is leading strand. Okay, S2 is lagging strand. Okay, so leading strand is copied in the same direction as the unwinding process. Okay, so if we check this A, 3 prime to 5, this is S2. S2 goes in the same direction. Lagging strand is formed in the opposite direction. Okay, of the unwinding process. That is why it is formed by discontinuous replication. So S2 is in the same direction, and S2 is lagging strand. So anywhere you see S2 in the same direction as the unwinding process, that is wrong. Okay, which other place we have S2 in the same direction? Okay, right, this is going in the same direction. Okay, so that eliminates A, right, and B. Okay, and if you come to D, you see, yeah, it's going in. Can you see? You see S2 here is moving in the opposite direction, right, on the winding process, both in C and D. So which one is the answer then, right? So don't forget that if you check C, this is 3' to 5'. Okay, this is 5' to 3'. So which one, right, act as a template for S2? Okay, right. So which one? Of course, we are moving. Of course, it's going to move in the opposite direction to the unwinding process. Okay, so you see that is moving in this direction, right? It's moving in this direction, and then this is moving. Of course, you check this one. This is also moving in the same direction, but you see in this case here, you see that this is moving in opposite direction. This is moving in opposite direction. So that's S2, lagging strand. Is that okay? So this is lagging strand. Okay. So this is also lagging strand. All right. So this is lagging strand. All right. So if you check it, you see that 5' to 3', 5' to 3', all right, is used to code for lagging strand. While 3', all right, to 5' is used to code for leading strand. So that's why I think C should be the answer. All right. Yeah. So which diagram correctly shows the structure of a piece of DNA? All right. DNA. Of course, DNA is double-stranded, but pause the video and tell us what you think. Pause the video. Tell us the answer. So, but you see, DNA doesn't have uracil. So A has uracil. This is uracil. What DNA has is thymine. Okay. So that eliminates option A. Anyway, see uracil, that's eliminated. Option A is limited because of that. Now DNA strands move in anti-parallel. So if you check B, they move in parallel form. So that eliminates it. So we have C and D left. Do you get? But if you check C, you see that cytosine pairs with adenine. Not here, but that's wrong. Cytosine should pair with, it forms triple bond with guanine, not adenine. Okay, that eliminates this. But you check the, yeah. So anti-parallel, you can see this is moving from here to here. Okay, 5' to 3', this is 3' to 5', and then there is correct complementary base pairing. So I think D is the answer, guys. What do you think? Yeah, let's run. Bacteria were grown. Let's roll it quickly. So bacteria were grown in a medium containing heavy isotope. After several generations, all of the bacteria DNA contains 15. Some of these bacteria were transferred to a medium containing the common isotope of nitrogen-14. The bacteria were allowed to divide once. The DNA of some of these bacteria was extracted and analyzed. This DNA was all hybrid. DNA containing equal amounts of N14 and N15. That's hybrid DNA. All right. The remaining bacteria were left in the medium with N14 and allowed to divide one more time. The DNA of some of these bacteria was extracted and analyzed. What is the percentage of the hybrid DNA? Okay, so like you to work on this, right? So pause the video and work on it, please. Pause the video and work on it. So guys, look at it. So if this is the original DNA, after so many years, they were all N15. Is that okay? N15, and then the DNA, the bacteria were transferred into N14, right, medium. So in N14 medium, the two strands will separate. Each of them will be used as a template, right? Remember that this medium now contains N14. So the newly synthesized DNA strand will be made up of N14. So this is the first division, okay? So it's the first division here, a new strand here, this is N14. Is that okay? A new strand here, pumps up. This is N14. Do you get? Yeah. So this is the hybrid. So the hybrid here is 100%. Do you get? Yeah. Hybrid here is 100%. You can see. But they said they're allowed to divide one more time. One more. So each of them now each of them will form a template. So divide one more time. Let's see the percentage of the hybrid. So this is going to separate. Okay, you can see the two template strand of the first DNA molecule separate because they are going to be used as templates. So these two are separating. This is going to separate to So we're going to have N15 here. The strand here is N15. Pardon my annotations, okay? Now, of course, separating this is going to be N14. So each of them will be used as a template. So this will be used as a template to form N14 because they are growing in a medium that has N14. So N14 will be used, all right, to form a new template, all right? Good. So this is N14, all right, each of them. So you can see that's N14. So the question is asking us is that what is the percentage of hybrid DNA after second division? So this is the first division, this is the second division, all right? So hybrid out of 4, how many hybrid do we have? Remember that this is hybrid. So out of 4 DNA, 2 are hybrid, this and this are hybrid. So that means 2 over 4, right? That's half of it, all right? Half of it, okay? Half of 100% is 50%. So you see that every generation it will decrease by half. So this is going to be 50%. If we go third more time, it's going to be 25%. Is that okay? For the hybrid. So after the second division, it's going to be 50%. So this is the answer. Is that okay? Yeah, but if they say third, it's going to be A, 25. But this is second, all right? All right, let's go to the next question. Which statement correctly describes the process of transcription? Yeah. So which of them? So make a decision, guys. Which statement correctly describes the process of transcription? So mRNA is decoded by a ribosome. This is transcription. Transcription is synthesis of mRNA, okay? Using DNA as a template. So mRNA is decoded by a ribosome to produce a specific amino acid chain. The mRNA is not decoded by a ribosome. So it's decoded by a tRNA, okay? So this is wrong. Number 2 says a section of DNA is converted to RNA by RNA polymerase. That's it. So that section is called gene. That's what happens in transcription. Gene is copied and converted to mRNA. Okay? Now the third one says tRNA transfers amino acids to ribosome. Okay? tRNA carries amino acids to ribosome. So tRNA transfer, of course, it transports amino acid to ribosome so that there can be arrangement, right? Right? So they say which statement correctly describes the process of the transcription. Remember, we are working on transcription. You see, this happens in translation. It doesn't happen in transcription. Do you get? So that's wrong. This is correct, but it happens in translation, right? This, all right, of course, also is in translation, though what decodes it is tRNA, but it happens in translation. And what we're interested in is transcription. So I think 2 only is the answer. Yeah, let's go. Sickle cell anemia mutation. Let's look at that. Sickle cell anemia is caused by mutation in one allele of the gene that codes for the beta globin polypeptide of hemoglobin. The diagram shows a sequence of bases in a small section of template strand of DNA for both HbA, that's normal allele, and HbS sickle cell allele, beta globin alleles. So these alleles, LE, this is not correct spelling, it's a typo. So this is for normal, okay, and this is for abnormal. All right, what do they want from us? Both the polypeptides for this HbA and HbS are the same length. How will the mutation in the allele result in the production of altered version of B globin polypeptide? How will it, okay? Yeah, so pause the video, it's your turn. I'd like you to deal with this, okay? Fry this fish, right? Fry the fish. Solve the question, guys. All right, so let's roll through. Are you ready? So come with me, let's fry this fish. So the first question said, how will the mutation in allele result in production of altered version of beta globin polypeptide? Remember, the rule is that once there's a change in base triplet, it's going to affect, the amino acid sequence. So it affects the codon on mRNA and affects the anti-codon on tRNA. Okay, that affects amino acid sequence, which is primary structure. Okay, look at this. So A says a tRNA molecule with anticodon GUG will form hydrogen bonds with altered codon on mRNA. Okay, let's see what altered first. Okay, guys, let's see whether this is correct. Okay, let's see where the alteration occurred. Okay, that's where it occurred. All right. You can see that. Let me take it. Okay, yeah, you can see that. So look at this. So this is the same here. No alteration. First base triplet, second base triplet, ACT, no alteration. All right, CCT, third one, no alteration. We want to find out where it occurred. Now the fourth one, it seems there's a problem here. So there's alteration. So adenine, right, is replaced using thymine. You can see instead of A, we have T here. Is that okay? And of course, if you check the rest of them, no alteration here. See? No alteration here. All right. So that alteration, instead of GAG, all right, we're having GTG. So this GTG is the DNA version of it, right? The anticodon version of it is the same as that of the DNA version of it. The only change would be that instead of having thymine, we have uracil, because tRNA does not have thymine. So this is going to give us, okay, the tRNA version of this altered form. Instead of GTG, is going to be GUG, okay? Because the base triplet of DNA is similar to the anticodon of tRNA. Why? Because both of them are complementary to mRNA. Is that okay? All right. So I think this A is correct. So why is B wrong? So all the amino acids coded for after mutation will differ from those, not all amino acids, because it's only one part that is changed, okay? All of these, they are the same here, the same here. So this is wrong. Okay. C says mRNA transcribed from HBS allele will contain the codon CAC instead of CTC. All right. Now, can you see? Now this mRNA here, GAG, is supposed to be C, right? T. It doesn't have T, so CUC. So mRNA cannot have T, so this is wrong. It's wrong. All right. So if you check D, the ribosome will be unable to continue translation of HB after the alter. No, it will continue because there's no stop codon there, guys. So this is wrong. So the answer is, of course, A. So let's go to the next question. Which statement describes the structure of ATP? All right, pause the video and try it. ATP, adenosine triphosphate, okay? So it's a DNA nucleotide with 2 extra phosphate. Now, ATP is not a DNA nucleotide because it doesn't have deoxyribose. It has ribose. So this is, anyway, cDNA is wrong because ATP has ribose as its pentose sugar. Okay, yeah, but it shares ribose with RNA. So it's an RNA nucleotide with 2 extra. Yeah, it has 2 extra. So that's why instead of 1 phosphate group, it has 2 extra, making it triphosphate. So this says it's an RNA nucleotide with 3. No, it doesn't have 3 extra. Okay, so it has 2 extra, and that is why C is the answer. All right, rifampicin is an antibiotic. Okay. So read with me. Antibiotic to treat tuberculosis. It works by inhibiting RNA polymerase in bacteria. So what's the function of RNA polymerase? Transcription. Okay. Which processes are directly inhibited by these antibiotics, guys? So pause the video. Which ones are directly inhibited? RNA polymerase, okay, catalyzes transcription. Transcription is part of protein synthesis, right? Oh yeah, so I've already told you the answer. So DNA replication, no, because it's not catalyzed by RNA polymerase, catalyzed by DNA polymerase, right? Enzyme synthesis, yes, because enzyme synthesis involves transcription. ATP synthesis, no, because this occurs, all right, it doesn't require RNA polymerase. Okay, so it's only 2 that is affected. So yeah, 2 only. The table shows DNA triplet codes for some amino acids. You can see that. Yeah, cute. So say the base sequence on the template DNA strand coding for part of polypeptide is shown. You can see that. Yeah. So say 2 mutations occur in this sequence during DNA replication. Which mutated template DNA strand would result in a shorter polypeptide? Okay. So which one? So pause the video, try it. So which one will result in a shorter polypeptide? Okay, pause the video and try it. Now, shorter polypeptide means that stop codon appeared earlier. So this is stop codon. Anytime stop codon appears in translation is terminated. So this is ATC. Okay, good. Anyways, it is ATC. And if you check here, is there ATC here? No. ATC here? No. ATC. This is ATC, right? Yeah, that's it. So this is it. This appears shorter codon. So anytime it appears, it will be shorter. That means this amino acid will not be added. GCA. GCA is actually arginine. Arginine will not be added. Right? Yeah. So there's no ATC there. So this is answer to give us shorter polypeptide, because each time it appears, it will stop translation. All right. The diagram represents a nucleotide containing thymine. Good. Thymine. So I'd like you to see, this is thymine. Which statement about nucleotides are correct? Okay. So which one is correct? Which one is correct, guys? Which statement about this nucleotide are correct? Which statements are correct? Okay. So pause the video and try it. There's a thymine is a pyrimidine. Yes, pyrimidine, because it's single. It has single ring structure. Yeah, a base pairing occurs with 2 hydrogen bonds. Okay, yes, because thymine forms 2 hydrogen bonds with adenine. Yes, 2 hydrogen bonds. Yeah, that's correct. The carbohydrate can be ribose or deoxyribose. Now it can be ribose because it's found. Thymine is only found in DNA. So thymine is only found in DNA. It's not found in RNA. So not in RNA, but in DNA. Do you get? So that makes that wrong. So we have A and B correct. Yeah, let's fly. Some of the events that occur during transcription are listed. So these are the events that occur. So bonds. Okay, yeah. So I say before mRNA molecule leaves the nucleus, which event occurred twice during transcription? Okay. Yeah. So I want you to pause the video and try it. So the first one said bonds break between complementary bases. Now these bonds are hydrogen bonds. Remember, if this is DNA double helix, okay, look at it, DNA double helix. During transcription, all right, a section of this DNA unwinds, and when it unwinds, okay, bonds are broken. Do you get? Bonds are broken. So hydrogen bonds are broken. Is that okay? And then, so this section is what we want to copy. See? So this is the first time the bonds break. Okay? And then, of course, DNA, RNA nucleotides start forming hydrogen bonds. They form hydrogen bonds, right? So hydrogen bonds are formed. So bonds form between the complementary bases on the template strand here. Okay? So, yeah. Then after that, sugar phosphate backbone are formed. So you see that RNA polymerase then joins them to form a primary transcript. Is that okay? So that actually happens, all right? So remember that this guy forms bonds with the complementary bases on the template strand. Is that okay? Yeah. So free nucleotide pair complementary. Yeah, that happened. I showed you to you. Is that okay? We're looking for the ones that occurs twice. Okay, yeah. But when this is done, the hydrogen bonds are broken. So they are broken first here when it's unwinding. Okay, so they are broken when mRNA is about to leave. Okay, so that break is twice. But remember that after transcription, DNA double is wind up. So in winding up, remember that the hydrogen bonds are formed twice. They are formed when nucleotides are pairing with their complementary bases. Remember, okay, if this is A, okay, the complementary base here will be C, will be U, right? So when they are leaving, this is the first time they are formed, okay? But when they leave, this unwinds. That unwinding means that hydrogen bonds are going to be formed again between the complementary strands of DNA. So two of them. All right. So this is correct. This is twice, right? This is not twice. That's not twice. So it is 1 and 2. Okay. The table shows DNA triplet code for one amino acid. You can see that. Okay. The base on the DNA template strand coding for part of DNA sequence is shown. 2 mutations occur in this sequence during DNA replication. 2 mutations. Which mutated template DNA strand would result in a shorter polypeptide? The one that has ATC. I think we'll solve this question. So solve it again. We'll solve it. It's repeated. So that's C. Okay. All right. AG codes for the sequence of amino acid in a single polypeptide. Hemoglobin consists of 2 alpha globin and 2 beta globin. Let's see. How many genes needed to code for a single hemoglobin hemoglobin molecule? So how many? So pause the video and try it. Remember that a gene codes for the sequence of amino acid in a single. So a gene codes for single polypeptide. Is that okay? Hemoglobin consists of 2 alpha and 2 beta. So this alpha, the 2 of them are identical. So they are going to be coded by 1 gene. The 2 beta are identical. They're going to be coded by 1 gene. So that means 2 genes will be coding them, right? All right, because the 2 polypeptide chains, each of them 2 alpha, identical to so all right. So 2 is the answer, right? Yeah, good. Okay, let's go on and look at this. You can see a single skin cell was isolated and transferred to a growth medium in a sterile petri dish. The petri dish incubated for 19 days. During this time, the number of skin cells multiplied as a result of repeated mitotic divisions. One of the chromosomes in the nucleus of each skin cell has a telomere that contains many repeats of base sequence. Let's repeat on each of the days 4, 8, 12, and 16 of the incubation period. Okay, a single cell was removed from the petri dish. The total number of bases in the telomere of this chromosome was determined for each cell. Each of the 4 cells had a different total number of telomeres bases for this chromosome. So you can see that. Okay. Now, what was the total number of telomere bases, okay, in the chromosome from the cells that had undergone the most mitotic division? So like you to fry this fish, fry it, okay? So you can see, guys. Yeah. So what do you think is the answer? Remember, guys. Remember that we always say, right, that the length of telomeres, which is determined by base, okay, length of telomere, is affected. So length of telomere, LT. There's a relationship between length of telomere and the cell division, number of cell division, okay? We say that the length of telomere is inversely proportional to cell division. So that means the more cell division, the shorter the length. So the ones that give us the most mitotic division is the one that gives us the shortest length. That's the one that has the shortest, right? Are you following? That's the one that should give us, all right? This is okay. So that's this is smallest, right? All right. This is smallest. Check it. It's not the smallest. It's the smallest. So that is A. So because the more, anytime a cell divides, the telomere depletes. In normal cells, telomerase enzymes that replenish it, all right, is inactivated, right? So the most mitotic will give us, all right, the smallest, all right, the shortest length of telomere. So that's why it's A. All right, let's go to the next question. A short piece of DNA, 18 base pairs. 18 base pairs long, was analyzed to find the number of nucleotides. Okay, uses each of the polar, right, in the nucleotide strands. So it says some of the results are shown in the table. All right, so I bring to you chocolate. How many nucleotides containing thymine were present in strand 2? So this is chocolate, guys. Enjoy it. Okay, enjoy it. Solve the question and tell me the answer. All right, don't you like this chocolate? All right, so look at it. I have 2 strands of DNA. Let's solve it. This is a strand of DNA. This is another strand of DNA. Okay, so this is strand 1. I'll call it S1. Anti-clean it. Strand 1. Yeah, let's erase that. Yeah. Okay, so stay with me. Strand 1, yeah, S1. Okay, and we have S2, strand 2. Okay, remember each of them will have adenine, of course, like they showed us. A is adenine, cytosine, right? What is it called? Guanine, yeah, and thymine. Is that okay? Yeah. So of course, because this is adenine, here is going to be thymine. Is that okay? Because they are complementary bases. Okay, because this is cytosine, this is going to be guanine. You know how it goes. Complementary base pairing. C goes, G goes with C, and this goes with A. See that? Good. So let's play, guys. Now they say let's play this game. Now for strand 1, we have 4 cytosine. Strand 1, okay, let's go. This is strand 1. Cytosine is 4 here. And if it's 4 there, automatically guanine here will be 4. Is that okay? Yeah. Still on strand 1, we have 7 thymine. So thymine is 7 here. That means adenine here with 7. Okay. Now if you check strand 2, we have 5 cytosine. This is strand 2, 5 cytosine here. Okay, that means that guanine will also on the first strand will be 5. So the question says how many nucleotides are containing thymine? Okay, so let's see. Okay, how many? There's nothing for this adenine and thymine here. Is that okay? So let's find it. Remember that we are told 18 base pairs. So let's add these base pairs. This is 4 base pairs, 5 base pairs, 7. So the base pairs we have here is 4 base pairs plus 5 base pairs plus 7. Okay, so what would that give you? That will give you 16, right? 16 base pairs. Now how many base pairs are left? So you say 18, all right, which is total number of base pairs minus 16 that will give you. What we're going to have left is 2 base pairs. Okay, that means we're going to have 2 adenine here and 2 thymine here. And what we are looking for is thymine in strand 2. So thymine in strand 2 will be 2 sit here 2. Okay, so you can watch this video again, okay, replay it, but the answer is 2. All right. The diagram shows a strand of DNA and mRNA during transcription. You can see that. Which row correctly identifies, just one identified, that's per typo, 1, 2, and 3. Which row identifies 1, 2, and 3, guys? Yeah. So which row identifies it? All right, I'd like you to do that. Now, 1 is cytosine. Cytosine is a pyrimidine. Okay, so that eliminates A and B. Okay, now 2 is a pentose sugar, which is a deoxyribose. See, so this is where C is eliminated. So D is eliminated. So we have C as the answer. All right. Well, no. Wait. We're looking at transcription. Yeah, transcription. Yes, transcription. So that means 1 is DNA, 1 is RNA. Yeah. So pyrimidine. Now, if you come here, we're not sure of which one, but this will decide. All right. So guanine forms 3 hydrogen bonds. So that 3 is 3 hydrogen bonds. Yeah. So actually, C is wrong. D is the answer. Do you get? Because they say transcription, that means that this is a DNA nucleotide pairing with RNA nucleotide. You know their bases. Do you get? Okay. So pyrimidine 2 is going to be ribose, right, or deoxyribose, but of course 3 should be 3 hydrogen bonds, and this meets all the requirements. So C, D is the answer. D meets all the requirements. Okay, D is the answer, please. Yeah, 2 students were when discussing the involvement of DNA and RNA transcription. Translation. Let's get it on. Okay. Translation. Student 1 always stated correct fact. So 1 is always correct. Student 2 gave further information which was sometimes correct. That means sometimes it wasn't correct. All right. See that these are the facts. So the question says which further information given by student 2 is correct. All right. Which one is correct, guys? So pause the video and try it. Pause it and try it. Is that okay? Fry this fish. Now let's go, guys. Now 1 is always correct, so we're going to focus on 2. So 1 says a length of mRNA is 747 nucleotide long, including stop and start codon. Okay, so now 2 says this mRNA can produce a polypeptide that is 249 amino acids. Okay, let's check it. Stop codon doesn't code for anything, so if you minus 1, okay, from 747, you're going to have 746. That is the length of nucleotide. Okay, so if you want to know the number of amino acid form, you divide it by 3. Is that okay? Because 3 nucleotide gives us 1 amino acid. When you divide it by 3, so if I have 7, 4, I'm pressing my calculator, precious, divided by 3 gives me what? It gives me, okay, yeah, okay, 248. Of course, it's not up to 249, right? That 248 point something, 248, right? 248.66, 248, okay? 0.6, right? 6 something. So this says 249. So this is wrong. Is that okay? Now sometimes the first amino acid may not be part of the protein, okay? But you know the first codon, that as the start codon may not be part of the protein because of course the start codon codes for methionine, but you know it has to be there because it starts codon so that translation can begin. Is that okay? So this is likely not going to be 249. Is that okay? All right. Let's look at number 2. Adjacent mRNA. Okay, this is what I'm going to focus. They say adjacent mRNA codons of AAU and CUG binds to complementary tRNA anticodon. Okay. They say there is a total of 14 hydrogen bonds formed between these 2 codons and their anticodons. All right. Remember, let's check it. This A will form 2 bond with U on the anticodon. This will form 2 hydrogen bonds with U, 2. and it's going to form 2 with A. That's 6 already. Now C will form 3 hydrogen bond with G. Okay, that's 9. U will form 2. 9 plus 2 is 11, and then this is going to form 3, so that's 14. So this is correct. If you add up this, so that is correct. It's 14 hydrogen bond. So this is correct. Okay, so let's check the second one. RNA polymerase catalyzes the formation of mRNA from the template strand of DNA. So during translation, an RNA adenine nucleotide will pair with a DNA thymine. Okay, now during translation, see what I said during translation. DNA is not involved in translation. This is wrong. Now 4 says a DNA adenine nucleotide is structurally different to an RNA adenine nucleotide. Okay, the difference is in hexose sugar. They don't have hexose sugar, they have pentose sugars. This is wrong. So it's only 2 that's correct. Okay, yeah, 2 only. So the answer is C, which is 2 only. Is that okay? All right, let's go to the next question. Which letter in the key describes uracil? Pause the video and try it. Which one describes uracil? Now uracil is not a purine. Is single ring structure. So it's not a purine. So it has a single ring structure. Yes. So that's C. Okay. So that is it. When a gene for protease is activated, which nucleic acid will be formed? Pause the video and try it. But anytime a gene is activated, right, the gene will undergo transcription. That's okay. Switch on. Okay, and the product of transcription is not DNA, it's mRNA. It's not this. Okay, it's not this. It's mRNA, right? That is the product of transcription of a particular gene. You know, most genes. Is that okay? So that's it. A piece of DNA molecule contains 84 base pairs. The table shows the number of adenine, okay, and of course the bases in one or both of the DNA strands in this piece of DNA molecule. Let's check. You can see that. How many guanine bases are present? How many guanine bases are present? So who is going to fry this fish for me? How many guanine bases are present? All right, please pause the video and fry this fish. Solve the question. Okay, just like we have always done, guys, watch this. This is 2 strands. This is strand 1, okay, and then we have strand 2. Okay, let's lay it out, and then this is strand 2. Now strand 1 has adenine. You can see you have adenine here. Okay, yeah, so the adenine here is, of course, we also have cytosine. So adenine here is 28 for strand 1. You can see that. But don't forget that adenine pairs with thymine. All right, adenine on strand 1 pairs thymine on strand 2. So it's going to be the same, 28. All right. Now cytosine in strand 1 is 15. So it's going to, of course, pair with 15 guanine. So guanine here, because they're complementary bases, is actually 15, guys. Yeah. So they say how many guanine bases are present in this piece of DNA molecule? How many guanine bases, okay, are present? Okay. So what we do is let's find the total base pairs. If you say 28, that's the one that are present. 28. Remember that we're not mentioned the guanine present in strand 1. Nobody mentioned the thymine present in strand 2. Okay? Nobody mentioned it. And nobody mentioned okay. So of course we're gonna have C here, we don't know, right? We're gonna have A here, right? So the question says how many guanine bases are present in this piece of DNA. Okay, let's go, guys. So if you add, right, 28 base pairs, right, plus 15, what will it give you? Okay, 28 base pairs from plus 15, what will it give you? That will be what? Punch your calculator. So that will be, remember that we're not done. So adenine in strand 2, that is adenine in strand 2 was 23. Is that okay? Yeah, we didn't add it. So that means thymine here will be 23. Okay, so let's add it. So plus 23. 28 plus 15 plus 23. What will it give you? So bring your calculator. 28 plus 15 plus 23 gives you what? 66. Alright, so we say 88. Now what we have is 84, right? Minus 66. 88 is total base. So let's minus it, so I can find guanine here. We don't know this guanine here, okay? Whatever it is, okay? This guanine here, we don't know it. So we can add this guanine here and this guanine here because we're looking for the total of guanine, guys. Okay? So let's roll it. So 84 minus 66 will give us what? That will give us 18, I think. Yeah, that's 84 minus 66. So okay, that's 18. So we have 18. So that means we're going to have 18 base pair. So that means this 18 base pair is going to be 18 guanine here and then 18 cytosines. Okay, so everything is complete. So now I have 18 guanine here plus 15 guanine there. Do you get 15 here? Is that okay? That gives me the total. That's how many guanine bases are present in the entire DNA, right? So I will say 18 plus, right? 15. That gives you 33, okay? All right. So this plus this, right, gives us 33. And that is B. All right. So the mRNA sequence of the 3 stop codons are shown. You can see that. Which mutation in the template transcribed strand of a DNA sequence that codes for polypeptide would cause a translation to stop prematurely? Okay. So I want you to. So this is sequence of 3 stop codon. That means this is the first one. So if this appears, it's 2 stops here. This is the second one to appear. This is the third one. Okay, so if this one appears, that means there's going to be a much more shorter polypeptide. But if this appears, okay, there's going to be, okay, longer bits. Okay, all right, there's a which mutation. All right, if you check this, which mutation, let's check this. Remember that this is tRNA, mRNA. Okay, so let's check this. If you convert this to, all right, to mRNA, this is going to be U, A pairs with U, U, A, all right, UAG. So UAG, yeah, that's the second stop codon, all right, in the sequence. Okay, let's check the next one. AC change to ACA. Now ACA, that would be, okay, that would be U, G, U. Do you have UGU there? No. So let's check the other one. ATA. That will be U. Okay. A. T pairs with A. AA. Alright. This is AA. UAA is the first one. Okay. To appear. Now change to TAG. This is going to be U. AC. Is there UAC there? Nope. So there's no USC. So we have these two showing up. So the one that appears first, the first stop codon is this UAC, which is that's UAA, which is option C. So two of them form stop codon, but the first one, the sequence is UAA, and C gives us UAA first. Okay, AU, okay, TATA. So the answer is C. All right, guys. Thank you for following up. This is where we are stopping the video. Now, I want you to get ready for our next video on topical plant transport. Okay, see you later. Bye.